This is part 2 of the chapter 15 lecture notes. Um, so what you're looking at here is an illustration of the initiation step of transcription. You can see that the first step is that transcription factors. That's what TF stands for in some of these molecules that you're going to see. Transcription factors recognize the promoter. So the promoter includes that TATAA. I think I said that right. TATAAA. That region, which is 30 base pairs down from the transcription start site. So that's why it says minus 30. So transcription factor D recognizes that region of the promoter and binds to it. And TBP actually stands for Ta-Ta binding protein. T-A-T-A binding protein. That's what TBP stands for. And that recognizes this portion of that sequence. And so it comes in and binds. And then you have other transcription factors that join in. And once all the transcription factors and the TATA binding protein, all of those have attached, then transcription is initiated, and the RNA polymerase II can start adding nucleotides to the DNA strand. And this is just an illustration of elongation. Messenger RNA is synthesized just like DNA is replicated from the 5' to the 3' direction. The FACT complex reassembles the nucleosomes as polymerase synthesizes the messenger RNA. So this is just a complex of molecules that wraps the DNA back around the histones. If you remember, that's how DNA exists as chromatin. It's DNA and proteins called histones. So that's what it means when it says reassembles the nucleosomes. Nucleosomes are the DNA plus the histones, histone proteins. Okay? And then the termination. RNA polymerase requires a termination protein that recognizes a specific 18 nucleotide sequence. That's RNA polymerase I. RNA polymerase II terminates by forming a hairpin loop. And RNA polymerase, that's 3, I'm sorry, I said 2. I mean, you would think it would be 2. And then RNA polymerase II transcribes 1 to 2,000 nucleotides in excess of the gene template, which are removed during messenger RNA processing. So these 1,000 to 2,000 nucleotides, let me ask you this. Do you remember, would they be called exons or would they be called introns? And the answer, of course, is they would be called introns because those are the pieces that are spliced out during messenger RNA processing. All right, after the splicing out of the introns, or actually before, sorry, got the order wrong. So during messenger RNA processing, before the introns are spliced out, there is a 5-methylguanosine cap added to one end of the messenger RNA, or 5' added to the 5' end, and then a poly A tail added to the 3' end of the messenger RNA. Once the cap and the tail are added, the introns are spliced out, and we've already looked at this. This is just the same. Here's your 5' methylguanosine cap and your poly A tail, and you can see that the introns have been spliced out. Messenger RNA splicing involves removal of introns. The splicing process is catalyzed by spliceosomes, and these are composed of proteins and RNA molecules called snRNAs. So the spliceosomes are responsible for splicing or cutting the introns out and splicing the exons back together. rRNA stands for ribosomal RNA. Or it's usually written little r RNA. And then tRNA is transfer RNA. Which is usually written little t RNA. So these molecules are processed with these 4 steps: intramolecular cleavage, splicing, methylation, and the chemical conversion of nucleotides. And this is a space-filling model of a transfer RNA molecule that adds the amino acid phenylalanine to a growing polypeptide chain. It contains the anticodon AAG, which is going to be complementary to the codon on the messenger RNA, UUC. And then it's going to attach phenylalanine to the amino acid phenylalanine is attached to the other end. So in a transfer RNA molecule, the amino acid is attached at one end, the anticodon is attached at the other end. And that transfer RNA knows exactly where to go because that anticodon is complementary to the codon on the messenger RNA. So it will bind with it. Protein synthesis, which is basically what we're talking about in this chapter. Protein synthesis decodes messenger RNA to produce a polypeptide. This is what we're talking about now is the translation part, okay? Polypeptides are formed, and polypeptides, remember, are long chains of amino acids that form and that are later formed into complex shapes, and then they're called a protein. So a polypeptide is really like an immature protein, okay? So they're formed when the amino group of one amino acid forms an amide bond with the carboxyl group of another amino acid. That we normally call that a peptide bond, okay? So proteins are formed when amino acids join with other amino acids, and ribosomes catalyze these reactions, the attachment of amino acids to other amino acids. This is showing you how a peptide bond forms. Water is removed by removing the OH from the carboxyl group of one amino acid and the H from the amino group of another amino acid. And after the water is removed, the carbon and the nitrogen then form a covalent bond. That's called a peptide bond. The translation process, translation is also called protein synthesis, begins with initiation and then it has elongation and termination steps. So same 3 names as transcription. Initiation consists of messenger RNA attaches to the smaller subunit of the ribosome. You're going to see that a ribosome has a large subunit and a small subunit. So messenger RNA attaches to the small subunit of the ribosome. The start codon AUG, a transfer RNA with methionine attached. Methionine is always the amino acid that AUG codes for. So it's always the first amino acid in the protein. So a transfer RNA containing methionine attaches to the AUG start codon. And then the larger subunit of the ribosome comes in, and the messenger RNA is attached like that. The larger unit comes in, and it's I think it's sort of like a sandwich, with the messenger RNA being in the middle. And then the elongation occurs with the transfer RNAs move in with the appropriate amino acid. The amino acid chain grows, The enzyme involved here is called peptidyl transferase. And termination happens when a stop codon is reached. And there are 3 stop codons that do not code for amino acids. They just code for stop making protein. And then the amino acid chain is processed. And it will, in eukaryotic cells, the amino acid chain will move to the endoplasmic reticulum to be further processed. This is an illustration of the translation process. And you can see here the transfer RNA has brought in methionine to the start codon. And you can see that messenger RNA has attached itself to the small ribosomal subunit. And then the large ribosomal subunit comes in, and transfer RNAs continue to bring amino acids to the correct spot. Now, once this one comes in, it's gonna attach to the methionine. The phenylalanine will attach to the methionine, and then this transfer RNA will eventually leave. Or you can see this one. Okay, so the UAC, this transfer RNA that's got methionine on it, that one, after it adds the methionine to the protein chain, it can now leave. It can go back into the cytoplasm and find another methionine and carry it to, make a new protein. It can just be reused over and over. But after the transfer RNAs have attached their amino acid, they leave. So these sites here are called the A site, the P site, and the E site. And the E site is, I think of it as exit. The P site is the permanent. I think of it as permanent. That means the amino acid has become a permanent part of the protein. See how phenylalanine is now attached to methionine. So it is a permanent part of the protein. And then I think of A as meaning the added site. This is where arginine is being added, but it hasn't become a permanent part yet. That's just my take on it. Oh, wow. Okay, so that's it for this chapter. Thank you.